So you're ready to dive into the incredible world of biotechnology? That's awesome. But, behind every single game-changing discovery, there's a bedrock of safety that makes it all happen. So whether you're a student about to start, a new technician, or hey, maybe you're just curious, think of this as your guide to hitting the ground running the right way.
Okay, picture this. It's your first day. You've got the fresh notebook, you're excited to get started, but what do you do first? Well, before you even dream of touching all that cool equipment, you've got to understand the space you're in and how to move through it safely.
Before you pick up a single pipette, you really need a mental map of the lab. You see, most biotech labs are split into these specialized zones, and knowing what's what and where's where is absolutely key to keeping yourself safe and your experiments from getting contaminated.
First up, the wet lab. This is pretty much the heart of the whole operation. It's where you'll see all the long benches, the sinks, and that common equipment like centrifuges and shakers. This is going to be your main workspace for most of the hands-on stuff.
Okay, next is the media room. And the best way to think of this place is like the lab's own super clean, specialized kitchen. This is where you'll prepare all the food for your cells, the culture media, different solutions. And the number one rule in here? Preventing contamination. Because even one stray microbe can totally wreck an experiment.
And then you have the instrumentation room. This is where the really powerful high-tech gear lives. We're talking about PCR machines that can copy DNA millions of times over, and those ultra-cold freezers that are holding precious samples at minus 80 degrees. As you can imagine, this stuff requires some serious training to use correctly.
All right, so you've got the layout. Now let's talk about how to act. And these aren't just a bunch of random rules. This is the professional code, the habits that keep everyone, and all that important science safe and sound.
These might seem basic, but they are so critical. The lab is a dedicated space for science, which means absolutely no food, no drinks, no exceptions. And to prevent some really dangerous mix-ups, you have to keep your bench tidy and, this is a big one, make sure every single container is labeled. An unlabeled bottle is basically a mystery box of danger.
Now, I need to be absolutely crystal clear on this next one, mouth pipetting. You might have seen it in an old black and white movie or something, but in any modern lab, this is a non-negotiable, absolute never. You're literally risking drinking whatever chemical or biological agent you're working with. We have mechanical tools for this, like pipettes. We use them, always.
So why all these rules? Well, they all exist for a pretty good reason. To really get it, we need to talk about the potential hazards you'll be handling, maybe even on a daily basis.
You can basically sort lab hazards into two main buckets. You've got chemical hazards, which come from non-living things like acids or solvents. And then you've got biohazards, which, you guessed it, come from living things, like the bacteria we're trying to modify or other microorganisms.
Let's dig into those chemical hazards a little more. They come in different flavors of danger. Some things, like the ethanol we use to sterilize everything, are super flammable. Others, like strong acids, are corrosive. They can give you a nasty burn. And then some chemicals, like ethidium bromide, which is a special dye we use to see DNA, are potential carcinogens. You have to handle those with extra care.
Right. That's the million-dollar question, isn't it? You can't tell what's dangerous just by looking at a clear liquid. So how do you find out what you need to know to handle it safely?
Well, the answer is something called the Material Safety Data Sheet, or MSDS. Just think of it like a full biography or a user manual for every single chemical. It tells you everything you need to know, what it is, what the risks are, and exactly how to work with it safely. You should always check the MSDS before you use a new chemical for the first time.
Okay, I know an MSDS can look like this huge, intimidating document, but here's how you can get the most important info fast. Just focus on these 5 things. 1. Hazard identification. Just look for the big signal words, like danger or warming. 2. First aid. You need to know what to do if an accident happens. 3. Check the PPE section. That's personal protective equipment. And last but not least, 4 and 5, find out how to handle, store, and dispose of it properly.
So you know the layout, you know the rules, you know the hazards. Now, let's talk about your personal safety toolkit. This is your armor and your emergency plan. This is what's going to keep you safe.
Your first piece of armor, of course, is the lab coat. It's your first line of defense. It needs to be on and fully buttoned up anytime you're in the lab. And, super important, the lab coat stays in the lab. Never, ever take it home with you.
Next up, your eyes. This one is completely non-negotiable. Your eyes are irreplaceable.
A chemical splash can cause permanent damage in just seconds. And no, your prescription glasses do not count. You need proper safety goggles that create a seal around your eyes to protect you from every angle.
And the final piece of your armor? Gloves. They're a two-way shield. They protect your hands from whatever you're working with, and they protect your experiment from you. Just remember to change them often, especially if they get splashed, and always take them off before you touch things like a doorknob, your computer, or your phone. But look, even when you're super prepared, accidents can still happen. So what's the game plan when something goes wrong? Knowing exactly what to do beforehand is what keeps a little problem from turning into a huge disaster.
If there's a fire, you need to remember one simple acronym, RACE. It stands for Rescue Anyone Who's in Immediate Danger, Alarm by pulling the nearest fire alarm to alert everyone, Contain the fire by closing doors as you leave, and Evacuate. Get out. Your safety is always, always priority number one.
Now, and this is really important, you should only try to use an extinguisher if the fire is small, you have a clear way out, and you've been trained. If you do use one, remember another acronym, PASS. You pull the pin, aim the nozzle at the base of the fire, not the top of the flames, squeeze the handle, and sweep from side to side.
What if you get a chemical on your skin? Or worse, in your eyes? Don't panic, don't wait. Get to the nearest eye wash station or safety shower immediately and flush the area with water for a full 15 minutes. It's going to feel like a really long time, but it's absolutely critical to make sure that chemical is totally washed away.
Okay, so we've toured the lab, we've learned the code of conduct, we've decoded the dangers, and we've geared you up with a full safety toolkit. You see how all these pieces fit together? They let you do your job not with fear, but with real confidence.
You know, it really all boils down to this. Safety isn't some boring list of rules that holds you back. It's the professional foundation that actually makes real, amazing discovery possible. When you master these habits, you're not just keeping yourself safe, you're getting ready to do some incredible science. So go on, now you're ready.